So good morning, Olivier. Thank you very much for accepting our invitation. Welcome to our launch event. I would like to ask you a couple of questions, and I would like to start by asking you to tell us about your research field, and especially something about the work you are doing on COVID-19 recently.
Hello, thank you for the invitation. In our lab, we are studying SARS-CoV-2 replication. How does the virus enter cells? What happens once the cell is infected? And we are particularly interested in studying the cell-cell fusion process, because we observe that once the cell is infected by SARS-CoV-2, it may fuse with neighboring cells, forming large multinucleated cells, and this is this process that we are studying. So those are basic cellular virology experiments, and we are, on the other hand, also studying the host immune response to the infection. How do antibodies counteract viral replication? How do antibodies neutralize cell-cell fusion? So the use of the nanolife technology is most helpful to address those questions.
Thank you very much. So you mentioned the use of nanolife technology. Indeed, you own since a few months a CXA, and you're using it to look at COVID-19. So what does this instrument bring on the table differently from all the other instruments that your lab owns already?
So we set up the nanolife microscope within our P3 L3 facility. So this is a place where we can handle the virus. So it's very useful to use living cells and living virus to follow the impact of viral infection on the behavior of the cells. And we were very lucky to observe striking changes mediated by the virus upon productive infection, and that's precisely what we are currently studying.
Thank you. That's very interesting. And what do you think about the type of data that the microscope delivers compared to other instruments?
So our preliminary results are striking and amazing, because we have unprecedented details about the cellular organization, the organelles, the nuclei, the nucleoli, the cytoskeleton, the mitochondria, the lipid droplets, the Golgi and ER apparatus. So we are in the process of analyzing those preliminary results. It takes time because we are not familiar yet with the different techniques of analysis, but thanks to the collaboration with the nanolife team, we are making interesting discoveries.
Thank you very much. So that was very, very interesting to learn. I have one last question. So I heard that you have a nice data set that you are willing to share with us today. Is it true?
Yes, I'm happy to share those preliminary results. They are not published yet, so we are formally reproducing those experiments to validate those results, but at least it can give you a kind of overview of the impact of SARS-CoV-2 on the cell fate. And as you can see in the movie, the cells, once they are infected, will fuse and with the neighboring cells, as I mentioned. And it's very interesting to study how this fusion. It's very interesting to study the consequences of cell-cell fusion on the overall organization of the cell. We are particularly interested in the ER and the mitochondrial aspects and behavior within the cells.
Okay, thank you very much for being here with us today once more, and we are looking forward to see your results published very soon. Goodbye. Thank you.
Bye. Thanks for the invitation.